Authenticated volume of samples to the sample well.
Ito lang aking arco 13, correct?
Oo, 30.
13. So, ma-add to 13 na place in water. 13 UL from the place in water. The MPC well. 13 UL. Wala na yung kawa sa corner. Filter. Like, ano, anak sa iya.
Ikaw pa yun ito.
Ano ba ang 9:30? Wala ka mga pressure, ah.
Okay lang.
Mga dapat 10 na pa. Hindi ba? Wala. Pero dapat 10. Magalon. So, ano na siya, at least sa inyo, mag-practice na mo ba na sa makanan may time, ano yung time. Okay, isang first one sa COVID natin. Si Boss, hindi siya pwede kasi hindi ba? Wala na siya sa may hallway. Bilisan niyo, faster. Ha? Uy, transfer! As in, 32 yan. May muna, wala sa likod. Wala akong pressure guys ha, pero anong oras na? Kailangan makaroon kayo ng train. Then, a close. Always close. Get a frozen 0.2 ml cooler raw from the freezer and place your car number of spot na cold raw. Then,
Yes.
ang cooler raw. Gamay. Thank you. Report, text na lang na sir. Dali na. Tapos, pakas ng blank i-balance para mas kinuha.
Ano yung buka ko? Okay.
Oo. Or kaya mo siya. Hindi kaya mo mas hindi na ang purpose ng action para makulit. Kumuhusan ko kay master. Ang spinning down na para makolekta. Okay.
Okay naman, ma'am.
Okay lang siya. Sige, hindi ka na lang po spin down. Sige. Tapos naman lang sun short ako. Yan lang. Hey, sir, boy. Hindi ka ta.
Ayos na technique mo.
Here's your program. Contaminate. Malipat ang light.
Hi po. Hindi ah. Harap kayo. Look at Tias. Charot. Daw kami mapuli. Galing ang bot kong kaigo. Kawit ka.
Ikaw nga? Okay, I'm done. Ay, pero ma'am, okay. Tapos, sa dalawang step 1 class instrument, 26 well, quantitation, standard error, agent at man, sa standard. Okay, name is demo run 8. Return demo. Return demo 8. Okay. At saka ayusin ng mabuti. Tandaan, anak ka mo?
D3.
Okay. 2 Close.
Close gid mo na sunod.
Close kid, may mga hair ka nga. Click. Na. Mga abubot.
Watch. Okay, kag-answer girls, nakatay ang
Then next is your scrub suit. So kung naka-scrub suit.
So kung damo inyo master mix, so 20 bika sample ang inyo i-process. So pwede ka mo kagamit na ang centrifuge tools. So araw na.
Ini 50. Ang reverse pipetting just to make sure na right ang volume na dinadispense niya since wala siya bubbles.
So discard.
Then discard ang remaining. Una na din.
So So take out the samples in transport pouch.
Yung working area from clean to dirty lang. So depende kung right-handed or left-handed ka mo. Disinfect naman. Then, isa ito nga, primary packaging. So, cross-check ang label sa sample sa information worksheet.
Then I have 2 samples. So mapa na lang po dila. So tangan ko siya, label pa siya, master mix. MM.
Disinfect naman. Then, isa ito nga, primary packaging. So, cross-check ang label sa sample sa information worksheet.
Brown mask.
Dito yung mga master mix.
So dapat tinlak ka pa. Hanggang tinlak na po. Kita lang napit na.
May ari yung Dr. Extra galing wala lang click. I don't know if it's yours. This one kay Michael ni ato isa.
Okay.
Tapos GCP bukas. Pati, ibili ka mabalik sa Inside to inside.
Alam mo, Jung. Wala ka large. May mask dito.
Small. Extra small. Ito ang purple talaga.
Extra small. Okay. So, i-divide din yung protocol. Pagkatapos niya na lang. Tapos niya na i-done.
Okay, done.
Ikaw mo papilion ko kakay na. Nag-assign.
Decide.
Sige.
The big boss decide.
Oh, si Dr. Ma design ko.
Okay, number 1 for decon disinfection, 37. Sinong person 1? Decon disinfection, and then up to throwing of all reagents. 37 to 40. Sino person 1? Sino person 1? Sino?
Dr. Chi.
Okay.
37. Decontaminate the pre-amplification area by wiping the surface with 10% bleach,
Sige, dali lang, gid,
then
sir.
alcohol.
Then 41 to 44, ma-load master mix kag ma-close ang NTC.
Then let it stand for maybe 1 minute.
Okay, 1 minute.
And then after
Hindi na siya blanco.
inaara lagi lang yung picture ko.
Ito ang gawa, tapos after 15 seconds, gato, 5 to 10 seconds dire. Hindi
Like,
ka
mas
naman.
naman. Parang hindi ko naman kasi mo temperature ang balis eh.
Okay. Paano ko mabala na ako? Nasa?
Pakita mo, hindi mo. Ay, gwapo! Sorry.
Ikaw ang fan.
Mamim, ito naman lang. Mamim ka di
Gawin mo myself, Chad. Medyo mawudla na.
Thanks, Claire.
Ika na, kaya may...
Ano ba? Dali ka.
Ano na eh?
May...
Okay.
Ini ang swap.
Oo, munik
Okay. Ang breaking
Ang breaking point yun eh.
Kita ko man yung may ara, pero parang...
Pero depende siya per kit. Kiss may ara, may mark, kiss wala.
Okay.
So, break lang nila based
Oo.
sa kadakoon. Kadakoon sa
Based na
MTM.
Okay.
Oo.
Hindi nga daw something nga ipasudlon o gid ang hasta sa breaking point ang kunyari gid sulod sa ilong ko.
Hindi.
Hasta sa breaking point hindi man gali as ilong
Ang breaking point meaning, ang muna imo hasta dira mo siya udon para isulod mo siya sa container.
Okay.
Pero kung ang breaking point mo isubra man sa container, ma-adjust ko man kaya po.
Tapos,
Kung
ang
dako
mani-
siya bala tubig
tapos
for the
kung ang muna
container
ni
na.
imo VTM. So, ang VTM is naka-triple packaging na sa. So, first packaging yan ari may foam.
Okay.
Tapos din sulod sa ziplock.
Okay.
Tapos may ziplock pa din dito. Triple packaging.
Di ba isulod
Ay,
na
yata.
Kung true sample na na.
Oo, kung true sample.
Yung one sample, one
Oo. Na triple.
Usually, ini ng BTM cotton, they isulod mo din. Or may ziplock pa gali.
May
Isulod mo din.
Isulod pa na din.
Oo.
Okay.
Muni yapit sa container.
Kay kung
Pwede na nga wala
mag-receive-
ziplock.
Oo.
Tapos isulod mo
Oo,
sa din. Tapos
I think so.
ziplock.
Ah.
Pero ang nadumduman ko pag di ba may paraffin pag na din.
Kung mag-ano iban, may ginaparafilm pa nila. Depende, basta ang standard sa ayata, which is triple packaging. Kung ano man na ang ginapamutangga nila, basta triple packaging.
Pag may absorbent dapat.
Okay. Sige,
Okay.
So, everything na ginasulod, ginadisinfect.
Okay.
Isa-isa.
Ang centrifuge, sulod yun ha.
Question, another?
Maski ibang pa ng sample. Bitak din siya.
Ano yun, di ba? Fish me lang.
Siya lang alam, siya mo ano lang.
Ano lang ginabutan, dili lang?
Sa labi ko.
Lead
Relax na nga.
Ano, baka na lang.
Tapos po yung usual na is, for example, if mag-submerge ka,
So, ma-transfer na to.
Where are you?
Tapos doon
Di ba gabi, flight mo to nang?
Oo, tapos...
Kaka-delay, gali ang sempak, gabi.
Malalim yung malalang buwan. Pero okay lang kung sa labas sila.
Atras, anay, atras. No, direo, direo, direo. Okay, lights, camera, action.
So, hi and welcome to the sequencing room of PGC Visayas. So these two are our next generation sequencers, the new additional equipment here in PGC Visayas. So this is the iSeq 100 and that is the NextSeq 1000. So the two has the same principle when it comes to processing our samples. The only difference between the two are their capacities and their output that they can process. So iSeq 100 can process up to 20 samples and can have an output of about 1.2 gigabases. That's around in 8 million reads for paired end sequencing. While the NextSeq 1000 can process up to 384 samples and that's around 200 million reads and 120 gigabase pairs. So NextSeq 1000, because of the huge samples it can process, it will run around 29 hours for 300 cycles. While iSeq 100 can run up to 19 hours in a 300 cycles processing. So the main application of these two for now is the SARS-CoV-2 biosurveillance of the Philippines, where PGC Visayas is processing all of the samples here in Western Visayas for the detection of variants of COVID-19. Other applications includes whole genome sequencing, 16S metagenomics, whole genome sequencing of bacteria, sequencing, amplicon sequencing, and others. So, hindi ko na pala.
So here is our extraction room. So in this room, we usually do our DNA extraction and RNA extraction. Also, we are using this room for our DNA and RNA quantification. Dito na ang kata.
Action.
So this is our laminar flow. So this laminar flow, it keeps a control. So
Go.
this is our laminar flow. Our plate vortexer, our plate minifuge, centrifuge. Salat ng balkon, plate. So this is our laminar flow. Our plate vortexer, our minifuge, our vortexer, our plate minifuge, our refrigerated microcentrifuge,
Go.
our swing bucket refrigerated centrifuge, our shaker incubator, our semi-microanalytical balance, the gel electrophoresis system, the gel documentation system, and our thermal cyclers. Okay.
Camera action.
Hello everyone, now that you're done with the room 1 and room 2, we will now go to the third room here at the PGC Visayas Laboratories, which is the PCR room. On Thursdays and Fridays, it becomes the documentation and library preparation room for the COVID sequence. Let's go! So in this room, we have additional storage for our reagents. So we have -20, and then we have the pharmaceutical ref, which is a 4-degree refrigerator. So what we store here are our sequencing reagents. We also have this regular ref for our normal reagents aside from sequencing. So I will just introduce you to some of the big ticket items here in this room. So the first one is the Attune NXT flow cytometer. So it works to count your cells and to characterize your cells. So if you want to know if your cells are smooth or granulated, this machine will be able to tell you that. The next big ticket item we have is this guy. This is the 6-Studio Genetic Analyzer. If you've known about the Sanger sequencing, this instrument can perform Sanger sequencing for you. So here's an example of the electrophoregram produced from sequencing of the 6-Studio Genetic Analyzer. So just a short background on how it works. So it works by using capillary electrophoresis. So this is a dummy cartridge. So you will have 4 capillaries. They look like needles coming out. And then you will have a polymer here that will act as your gel. And then when it passes through the gel, your size fragments will differentiate and separate into different sizes. And a camera will be placed here to take a picture of those fragments. And then it will show you later what those are. It will identify and take a base call, or it will show you if that sequence is an A, T, C, or G, and it will give you this final electrophoregram result. Next, we have our Agilent bioanalyzer. So it also has, it also uses the capillary electrophoresis technology. However, it is in a chip. So take your ordinary electrophoresis and put it in a chip. So inside, at the back of our chip will be capillaries, and our fragment or DNA fragments will pass through each capillary, and it will show you the size, the exact size of your sample. So if you want to take a closer look, it will have 12 wells.
Take a closer look.
Take a closer look. Next and last big ticket item in this room is the qubit 4 fluorometer. So it works by using the technology of intercalating dyes. So your intercalating dye will bind into your sample, and you can put your sample here in the qubit fluorometer, and it will show you the exact concentration of your DNA or RNA. And lastly, we have all the peripheral equipment that you will see in a mobile lab. So we have your pipetors and your microcentrifuges and the laminar flow. That's it for room 3. Let's go now to sequencing. Thank you and see you.
Action.
So here is our sample preparation room. So in this room, we usually do our extraction and so this is our sample preparation room. Before DNA extraction and RNA extraction, our samples are being prepared in this room.
Sige, tudlo.
So for storage of samples around -40 to -80, we use our ultra-low temperature freezer. So here is our liquid nitrogen generator, our ice maker, our Ridicule or water purification system. So this our two biosafety cabinets, our body-based fume hood, our incubator, our probe-type ultrasonicator. So for spinning down samples, the minifuge and then the vortexer, the plate centrifuge and the plate vortexer. This is our two autoclaves, our analytical balance, our spectrophotometer, multi-scan spectrophotometer, and our 4 thermocyclers. We also have our standard microcentrifuge and our refrigerated microcentrifuge. And lastly, this is our standard microcentrifuge, our refrigerated microcentrifuge, our vortexer, our hot plates, and lastly, our dry bath incubator.